For this lecture, we will cover Haemophilus, Legionella, Bordetella, and other fastidious gram-negative bacilli. Haemophilus species are small pleomorphic gram-negative coccobacilli. They are non-motile and oxidase positive. They are aerobic or facultatively anaerobic, and they require growth factors that can be found in blood, which include the X factor or the human factor or V factor, which is the NAD. And so these particular factors are found in chocolate agar. Sheep blood agar only has factor X, so the only way they would grow on the sheep blood agar would be if they were growing around a Staphylococcus aureus, and that's because that the staph does produce the V factor. And so because of that production, then the organism could get the X from the actual sheep blood agar, and the V factor that's produced by the Staphylococcus. So they would grow around that Staphylococcus. Our common pathogens are Haemophilus influenza and Haemophilus ducreyi. Haemophilus influenza it has two categories that are based on the capsule characteristics. So you have typable strands that produce the capsules, and there are 6 groups of that are groups A through F. The group B, or type B, Haemophilus influenza, is one of the most commonly encountered serious infections of this particular organism, and this is where we get the Hib V vaccine, so the Haemophilus influenza B vaccine. There are also non-typable strands, and these strands don't produce a capsule, and they are most often associated as the normal upper respiratory tract flora. Then you also have a biogroup of the Haemophilus influenza that can produce infections as well, so you will occasionally see this particular biogroup in culture. Haemophilus influenza is normal upper respiratory tract microbiota. It causes infections, especially in children, and so this is spread through droplets or through contact. It can cause meningitis, septicemia, pneumonia, and so these infections can be due to the encapsulated strand, or it could also be it's from encapsulated strands, so it can cause something as severe as the meningitis with encapsulated strands, and then something like conjunctivitis or ear infections with the unencapsulated strands. So these are gram-negative coccobacilli. They're non-motile, catalase positive, oxidase positive. And so when these organisms are cultured on blood agar, they will not grow unless there is Staph aureus present, because these organisms do require X and V factors. And so because they require X and V, then it needs to be a media that provides both of those, so the chocolate agar or a blood agar that actually has the Staph aureus on it. So if you look over here to the right, then you can see where this particular organism is doing what we call satellitism or satellite growth, where you actually have the staph streaked down the middle. If I can get my pointer right. So you have staph streaked down the middle. So there's a culture streak of Staph aureus. And then you can see the growth here around the edges where you have all those small pinpoint colonies that are very translucent, that are growing because of that V factor that is produced from that staph. And then here up top you have where there's an infant that has septicemia from this organism, and then it has transitioned to like a DIC where you have this disseminated intravascular coagulation. So you can see where there's some necrosis of that skin tissue on that infant. Haemophilus ducreyi, or Haemophilus ducreyi, depending on who pronounces it. This organism is transmitted sexually through the genital tract, and so this is not normal flora. And what it does is it causes genital lesions, and so you can see down here at the bottom, of course these aren't images of genital lesions because I'm trying to keep it a little PG rated here, but these are similar to the lesions that it would cause, those open wound type lesions. And so this organism is small pleomorphic gram-negative coccobacilli rods. And what you see with this one on a gram stain is a characteristic school of fish type appearance, where they kind of almost in clusters, but where these organisms are really close together, almost like you'd see like a school of fish. So you see all of those like pleomorphic type rods. So you can see this one here, you've kind of got two of them together where they're the coccobacilli, they're kind of elongated out a little bit. And so these organisms are non-motile, catalase negative, oxidase positive. They will grow on the blood agar plate because they do require the X factor, not the V. So not only will they grow on blood agar because it has X, not B, but chocolate agar as well because it has X and V. These organisms will produce a gray, yellow, or tannish type non-mucoid colony. But in order to identify this organism, most of these are going to be worked up through some sort of nucleic amplification, so some sort of STD probe where they'll actually do automated testing on these. So they would look for this particular organism based on that DNA type probe. With your X and V test, there are a couple of different methods. This particular method here requires the use of a nutrient type agar that does not have any sort of X or V in it, so there's no blood in it. And what you do is you actually do one of those lawn sweeps where you culture the organism over the entire plate, and then you place the strips down. And so each of those strips, the little pointed end, has one of the factors in it. So this top one has V, X, and XV. So this particular organism has growth around XV. And so this is considered a positive growth, or it requires X and V in order to grow. Then here in image B, we have V here to the side, X, and then XV here up top. And this organism grows around V and XV, so it requires V to grow but no X. And then here at the bottom, well, you got growth all over this entire plate. So this is considered a negative because it did not require X or V to grow. It grew whether it was there or not. But this is what you're looking for. You're looking for the growth in the presence of X and V, or X and or V. So with our Haemophilus species, we are doing that X and V test. And just a reminder that you can have some organisms that will demonstrate a positive satelliting on blood agar when they have staph present. And so it could just be that the organism would grow if you have just a staph from a culture. So you've got this that's grown and then it kind of zigzags out and you start getting staph colonies throughout, and then you would have your Haemophilus would actually kind of start pinpointing around that actual colony. Or you could actually do the satellite test where you would streak your Haemophilus or suspected Haemophilus across the entire agar plate, hoping for it to grow, and then you would come back with a streak of, get my colors changed here, you would come back with a streak of Staph aureus down the center, and that would allow that to produce that V. And then what you are looking for is that satellite where that yellow is a little hard to see, but you're looking for the growth of the Haemophilus just around it. Now, if it was to grow out here, then that would be negative. It would only grow in the presence of that staph streak. Haemophilus influenza will grow with X and V, so it does require both. So that would be like your positive satellite test. And then your Haemophilus dacrya would be, it requires just X, so you would see it growing around X and V and X. Legionella corridans is found as normal flora within the human oral cavity within the mouth. And this organism is transmitted through person-to-person contact, so this could be something like a bite or a fist wound. So a fist wound from a facial punch, and maybe they hit a tooth or something. Maybe like in this image here where that knuckle is infected. It can result also from the patient's endogenous flora, so the patient themselves could be exposed to their own flora, and it could disseminate and cause an infection somewhere else within that person. And so an example with this would be there's a group of bacteria that is associated with endocarditis, and so this particular organism is found within that group. And so for whatever reason, that infection, that organism could be dislodged from the patient's normal oral flora and then progress throughout the system to eventually reach and infect the heart and causing the endocarditis. And so it's also associated with aspiration pneumonia as part of like a mixed infection. So a culture from somebody with pneumonia could present with this organism as well as something else that's causing pneumonia. Legionella produces slender gram-negative straight bacilli with rounded ends. They are oxidase positive, catalase, urease, and indole negative. It is a facultative anaerobic organism. It will grow on blood, agar, and chocolate, but it does have a little bit of a slow growth. And what's unique about this organism is that it will actually pit or corrode the agar, and it will actually give off a chlorine type smell. And it does not grow on McConkey agar, but if you look at these images here, you can see where this culture of these colonies have actually kind of pitted away at the blood agar, so it gives a roughness to it, and you can see that in the bottom image as well. Legionella pneumophila is found, this organism is actually found in warm aquatic habitats. So you can find this organism in lakes, rivers, different marine water, moist soil, and it can also be found in man-made facilities. So this is the organism that is associated with like Legionnaire's disease, where it actually gets into like the ventilation system within your air ducts, and then it can be aerosolized and spread throughout the air system. And there's no person-to-person transmission associated with this organism. So these infections are acquired through the environmental sources, and so an example with the contaminated air sources through like your cooling ducts from stagnant water, then this would be transmitted through the air. The patient would inhale it. And these organisms actually possess genes that are able to trick the patient's cells, so like the white blood cells, and it will actually allow the cell will actually transport this organism into the cell, and then it will, inside that cell, it will replicate and multiply within that cell, and then it's excreted from that cell. So you can see an image here where the organism goes in, it actually replicates, and then it comes out of that cell. So at about 14 hours it has really multiplied. It also produces cellular components and extracellular products that impair the cell's ability to use oxygen or enzymes, so it actually causes problems for any of the cells that it enters into. Legionella pneumophila causes a pneumonia type infection, or also known as Legionnaire's disease. And so it does account for 1 to 5% of the cases of pneumonia. But if it is left untreated, it can have about a 30% mortality rate. It also causes Pontiac fever, which is a self-limiting, non-fatal influenza-like respiratory infection. And it also can cause an extrapulmonary infection, so it can infect wounds, encephalitis, or endocarditis. And those at risk include those that are immunocompromised, or 60 years of age or older, or have some sort of history of being a heavy smoker. These organisms are faintly staining, thin, gram-negative bacilli. They are obligate aerobic, and they are slow growers. And they do, again, require the iron and L-cysteine in the media to help them grow. So an example of a media that would be used would be a buffered charcoal yeast extract media that has the L-cysteine in it. And so at about 5 days, this organism is about 3 to 4 millimeters, and it is gray to white with a bluish green hue to it. It is glistening, convex, and circular. And they can also exhibit a cut glass type of appearance with this organism going on this plate. They're oxidase positive, catalase positive, and they also produce beta-lactamase. So this organism could be direct detected through a tissue sample, so material within the lung, some sort of biopsy from the lung tissue could be stained to look for this organism. Or you could look at the respiratory secretions and do an immunofluorescent antibody test to look for this organism. And you can also use other urine or body fluids to do some other rapid enzyme immunoassay testing. And then you can also do molecular testing through PCR or mass spectrophotometry. Legionella pneumophila is biochemically inert, so these organisms do not exhibit any sort of biochemical reactivity if you were to test them with any of the previous methods we've discussed about the testing done within the lab. These specimens should be cultured on a couple of different agars if you are suspecting this particular infection, or if you grew a culture and you suspected that this is what it is, or the gram stain demonstrated this particular organism, then you would do the BCYE agar, which contains the cysteine, as well as one that does not. The pneumophilia, the Legionella pneumophilia, will grow on the BYCE agar with the L-cysteine, whereas your other stuff, it requires this in order to grow. If it does not grow on the BCYE without the L-cysteine, then you would know that it was not Legionella. So these cultures are incubated 35 to 37 degree with humidity for up to 14 days, because it can take several days for these to grow. And then what you're looking for is those gray, white to blue-green, glistening, convex circular colonies. You can see that image up the top, where you're getting that little bit of a cut glass type look with that color pigment within those colonies. Bordetella protesus. This organism will enter the respiratory tract once it's inhaled, and then it binds to and destroys the ciliated epithelial cells that line the trachea and the bronchial tract. And so with these organisms, they have several virulence factors, one of which is adhesion. And so with this one, it will bind and actually start to paralyze the cilia, and then it will produce a tracheal cytotoxin. And then it also produces the pertussis toxin, along with several other toxins. And so these toxins will actually enter the target cells, and it will actually activate the paralysis or the destruction of the different cells. And then it also has the ability to overcome the host defense, and this is because of an outer membrane that it produces. So this particular organism, it mostly affects, it can affect different ages, but we usually do see this more or associate more with children, and what we call whooping cough. So with this, it starts out with very mild symptoms, maybe cold-like symptoms, and then it will progress to a severe violent cough, and this is where you get that whooping cough, where it has that whenever the child, once they cough and they're trying to breathe back in. And then it can go into a convalescent stage where it actually affects the lymph nodes. So adults and adolescents are recognized as the reservoir for transmitting this particular organism for this disease process. And then there are other species that have been associated with it, as far as Bordetella species, but those are usually just within the immunocompromised. And so over the years, there have been vaccines that have been developed that help to prevent this particular infection. And so those are your Tdap vaccines, and depending on the age that vaccine is given, depends on which one of those 3 vaccines that you actually receive. It is detected through a direct detection method, so something like fluorescent staining, or where you're looking for the actual antibodies from the nasopharyngeal material, or through PCR testing. And you can also do serodiagnostics, so when you're looking some of the different tests that are looking for the antigen-antibody relationship, and so those tests can also be used to help diagnose this particular organism. If we were to look at it in a microscope, it would be very small, faintly staining coccobacilli. They can be singles or pairs. And because it is difficult to stain, it can be enhanced with a 2-minute saffron O stain counter stain. These cultures do grow at 35 degrees Celsius with humidity, without carbon dioxide, for up to 12 days, so they can take a little while to grow. There's no growth on blood agar or McConkey, so these organisms are plated on a Reagan low agar. And when they grow on this agar, they produce a small, shiny organism or colony that can resemble a mercury drop. And then over time, they can be a whitish to gray with age. And so you can see an image of that there. They are oxidase positive, catalase positive, and they are non-modal, nitrate negative, and urease negative. Brucella species are categorized as a category B select agent. So these organisms are handled with caution in a biological safety hood, and they would be reported. And so this is a zoonotic disease, so they are transmitted through animals, and they do tend to localize in tissue that's rich in a particular sugar compound. And so these organisms are transmitted either through ingestion of infected unpasteurized animal milk, so like goat milk, through inhalation of the particles that have been aerosoled, through direct contact of infected animal parts, and through accidental inoculation of the mucous membrane, so introducing it to the mucous membranes. These organisms are actually ingested through the neutrophils, and once they're in there, they replicate inside those neutrophils, and then they are released from those neutrophils, which could be through destruction of the neutrophil itself. And so if these infections are left untreated, then they can cause granulomas in different organs, and they can also cause hepatosplenomegaly. This organism is identified through based on its characteristics of being a gram-negative coccobacilli or short bacilli. And so they stain very poorly on a gram stain, and they can be intracellular organisms, so you can see it inside of those neutrophils because, again, it uses those neutrophils to replicate. They are aerobic cultures, although you do have some that prefer the CO2 environment for growth. They will grow on a blood agar and chocolate agar, but they do require an extended incubation. They produce small colonies that are convex, smooth, translucent, and they can be, they're non-hemolytic, and they can also have a slightly yellow tinge to them. They do not grow on MacConkey. And so these organisms do better if they're actually cultured on a Brucella type agar that's specifically for these organisms. So those agars may be used if it is suspected. They are oxidase positive and catalase positive. And so usually these organisms are going to be detected through some sort of automation or PCR rather than its biochemical traits that you would see with some of the other organisms that we've previously talked about, where you work those up with different spot tests. Francisella tularensis is carried by rodents, rabbits, beavers. Humans become infected after they've handled the carcasses or the skins of these infected animals through inhalation of the aerosols. It can also be ingested with contaminated water, so maybe one of these carcasses was in the water supply. Through insect vectors, so there are different insects that can carry this, so getting a bite from that insect, and being bitten by one of these animals that are contaminated with this organism. And so it does cause tularemia, and the most causes of tularemia happen within South Dakota, Arkansas, Missouri, and Oklahoma. So this organism, after it has been introduced to the host, so through some sort of abrasion or skin puncture or perhaps a tick bite, then the lesion will start to develop at the site, and then it will actually start to progress to an ulcer type lesion. And so the lymph nodes that are near those lesions will become infected, and so they can become very enlarged and also become necrotic. So once the organism enters the bloodstreams, the patients become systemically ill, so they'll have like a high fever, chills, headache, aching, body aches. And then depending on how it affects the patient, it can range from very mild to mild and self-limiting type symptoms to very fatal symptoms. And so it does have different ways that it presents. So for example, glandular, which is is more common. You just have the large lymph nodes, the lymphedema. And then with this one, it's rarely fatal. And then, of course, you can have something like a conjunctivitis type infection. So you can see that eye where you've got those little glandular type lesions in the eye. And then it can also be something as systemic, where it causes the septicemia with that very high mortality rate. And with this one, you wouldn't have any sort of ulcers or the lymph nodes being infected. With this organism, it is non-motile, non-spore forming, strict aerobic. It does produce small, poorly staining gram-negative coccobacilli. And with this culture, it is slow growing, so it does take a couple of days to a couple of weeks to grow. And so in order to help this organism with its growth, you do want to use some sort of enhancement type media. And so it does grow better with with the presence of cysteine and iron. So you would look for a media that contains those to culture this organism if it was suspected. If it grows on MacConkey, it grows very poorly, but typically it does not grow at all on MacConkey agar. With chocolate agar, it would be pinpoint, small type colonies that would require a prolonged incubation in order for it to grow. And so they could be transparent or they could be mucoid. This organism is oxidase negative, and then it can be weak or a negative catalase test, just depending on the particular strand of that genus and species. And these organisms also have a negative satellite X and V test. So if you were to culture it doing a satellite test, that's where you run that strip of Staph aureus on the blood agar plate so that you would get both X and V, then this organism would not grow. Since this organism does not gram stain very well, it is of little use to actually use a gram stain to try to help identify it. And so this organism, again, like most of these that we've discussed, are typically going to be worked up with some sort of automation or direct detection method. And so when you look for like a particular antibody stain, a fluorescent antibody stain, to look for this organism, or something like mass spectrophotometry or PCR testing. And so this is a biosafety concern. Laboratory employees can be exposed to this if they were to handle this organism on an open bench top. So typically when reading culture plates, the tech does look at them out in the open. So if this organism was suspected, then they would need to move those plates to the biological safety hood in order to work them up to make sure that there's no aerosols created once you remove the top from this organism. Pasturella species are found within your domestic and wild animals, and they're usually transmitted to humans through some sort of close contact with the animal that is carrying this organism. And so these organisms are opportunistic, and so they require some sort of disruption to the host barrier or like a break in the skin in order to infect the host. And so this might be something like a cat bite, dog bite. Pasturella multocida, a subspecies multocida, is the most commonly encountered species that we find within the clinical specimens that are submitted for culture. This organism can cause a local soft tissue type infection. So here's an image here where perhaps it was maybe a dog bite. And then it can also cause chronic respiratory infections. So this might be in a person that's already had some sort of pre-existing chronic lung condition, and this person maybe was exposed, had a lot of exposure to an animal that was carrying this organism. And then you can also have a systemic type disease for something that causes something like meningitis, endocarditis, osteomyelitis, septicemia. And so the patient is actually inoculated through a bite or scratch. So you can see this a lot with your with veterinarians who may be exposed through dog bites or cat bites, scratches. And so you can also have infections that are associated with non-bite exposure, and less commonly infections can happen without any type of history of any sort of exposure. These organisms exhibit small, short, straight gram-negative bacilli, which have that bipolar staining. So you may have like little spaces in the actual stain of that rod. Growth on chocolate and MacConkey agar, and you have convex, smooth gray, non-hemolytic colonies. They can also be rough and mucoid, and they're described as having a mushroom type odor. There's no growth on MacConkey. They're non-motile, oxidase positive, catalase positive, indole positive, and urease negative. Rat bite fever is caused by Streptobacillus or Spirillum. And so these organisms are both gram-negative. Your streptobacillus is a bacillus, where your spirillum is actually like a spiral or a helical type organism. Your streptobacillus is a facultative anaerobic. It is non-motile, pleomorphic, and it causes rat bite fever. And your spirillum is aerobic and also causes rat bite. Streptobacillus is the normal flora of the upper respiratory tract of rats. And so this organism can actually be transmitted through a rat bite, or if you were to ingest any of the food that's been contaminated by this particular organism. And so it's typically found in unpasteurized milk and milk products. And it can also develop A-forms, and so this is like a bacteria without a cell wall. And what this does is it allows it to persist at the infection site. It actually allows it to be very viable at that site. So there are two types of infection with streptobacillus. You have rat bite fever, which is acquired through the actual contact with the rat, or through ingestion, which causes the Haemophilus fever. Symptoms can appear as early as 12 hours after exposure, and patients can experience an acute onset of fever, chills, headache, vomiting, joint pain, and then the patients can also develop a rash on their soles of their feet or on the palms of their hands. And so you can see that example here in this image where that skin is kind of pulled back. Almost looks kind of like that scalded skin syndrome, except this is going to be more localized to the hands and feet. The fever can persist for weeks or even months, and then it also can cause complications like endocarditis, septic arthritis, pneumonia, and even brain abscesses. So if we were to do a direct detection method, like through a gram stain, then you're looking for a gram-negative rod. And these can also appear as like long tangled chains or like a string of pearls. So you would see long chains of these gram-negative rods on that gram stain. Cultivation requires the presence of blood, Fastidous fluid, or serum in order for this organism to grow. So it will grow on a blood agar plate at 37 degrees with 5 to 10% carbon dioxide. And after 48 hours within that moist environment, you can start to see the growth. They're non-hemolytic colonies, and they can exhibit a couple of different appearances. And you can see an image here of one with that fried egg appearance, and where you have like a little bit thicker center where perhaps the yolk would be, and then the outside edges are thinner with a little bit more of a rough lacy edge to it. And this would be that L phase, those colonies from those bacteria that are in that L phase without that bacterial cell wall. These organisms can, if they were cultured within some sort of broth media, then you would see something that looks like fluff balls or breadcrumbs. So they would kind of just be little fluffy bodies growing inside of that media, like a crumb, a dissolved breadcrumb inside that media. They are indole negative, catalase negative, oxidase negative, nitrate negative, urea negative, and decarboxylase negative. They are non-motile, and they do produce H2S when you're using a lead acetate paper test. This is not a very common test though. Spirillum, it's very similar with the streptobacillus, and it is transmitted through a rat bite. The disease associated with this is Sudoku, and so this is a form of a rat bite fever in humans. And so the symptoms are very similar, with the main difference being that you don't develop the arthritis with this organism. You can have swollen lymph nodes, and they can be very prominent. The wounds will spontaneously heal, but you can have reulceration of those wounds, and it'll create the granulomatous lesion. And so the incubation period is a little bit longer than the streptobacillus rat fever, but also something that you can see is this rash that this organism will produce as well. Spirillum cannot grow on media, but if you were to do a gram stain on this organism, then you would see those spiral gram-negative organisms, and so these are called spiralochetes. So in order to detect this organism, you would it requires a little bit of an inhumane method where you actually use mice or guinea pig to inject them with this organism, and then you would test that actual animal looking for these spirochetes within that organism, within that animal. And so that would be a very long culture, because it can take like 1 to 3 weeks to actually recover that organism from that animal. So the best test method for this would be a serum antibody test. Bordetella is also transmitted zoonotic from animals to humans. And so humans will actually become infected naturally or incidentally. And once this organism comes in contact with the host cells, it will actually cause a proliferation of the microvascular endothelial cells. Some of the general characteristics include that it is a short gram-negative bacilli, and it is facultatively intracellular. It is very fastidious, but it can be cultured. If it was cultured on blood agar, it could take up to 45 days to grow. So obviously, you wouldn't want to be waiting on this test to figure out if this is what it is. It does not grow on chocolate or McConkey agar, and it is oxidase negative. Bordetella cattana causes a trench fever, and so this was associated with World War I soldiers that lived within the trenches, and so because of those conditions, they did develop this illness. And it causes a mild influenza-like illness that can have headache and bone pain associated with it. It also causes splenomegaly, and it can cause a rash. That rash is known as like a trench rash. And it can also cause bacteremia, endocarditis, issues with the lymphatic system, and bacillary angiomatosis. Bartonella Hensley can cause several different types of infections. However, it's most associated with cat scratch disease. And so with that, to diagnose cat scratch disease, you're looking for a history of animal contact, negative laboratory studies for the cause of any sort of lymphatic involvement, and you're also looking at a characteristic histopathological appearance of the lesion and a positive skin test using the particular antigen. So Bartonella can be directly detected in a tissue biopsy using the Warthin Steri silver stain. It can also be done by immunofluorescence or immunohistochemical techniques. And so there are 2 methods of culture that are recommended: inoculation onto fresh chocolate agar and co-cultivation with endothelial cell culture. Blood agar that is enriched with horse or rabbit blood is an effective supplement to cultivating this organism. Tissue and swabs can be inoculated onto late horse blood agar slants. And so once that is done, they would actually be incubated for up to 6 weeks to look for this organism. So obviously with these organisms that take a long time to grow, with automated methods now, different serology type testing methods, you're not going to wait for it to grow to figure out what's wrong with the patient. You're actually going to use some of these other automated test methods first.